Okay. I am so excited as your president to welcome you to ISDP 2021, and with half of you here in person, yay! And a full ton of people online, which is really exciting. So this hybrid thing is maybe a concept that will continue to happen. We are going to start our first symposium session, which is early crosstalk between the biology of stress and overnutrition. And I'd like to invite Brie Reid, who is the chair of this session, up here. And I'm going to sit down there.
Thank you. And is it possible to turn the table mic off? Because I think we're getting some echo there. It is? All right. Well, I guess maybe it's just resonant up here. I think so. I think we're all right. Can everyone hear just fine? Oh, perfect. Hi, good evening. I am so excited to be here and so thrilled and honored to be chairing this first symposium. I'm partially excited because this is the first time I've been in a room alone by myself since 2019, so that's been really thrilling. But I'm also just so excited to bring this symposium to all of you. The work presented here cuts across several domains and uses both human and animal models to bring forth insights on new avenues of research on stress and nutrition exposures across development. I also have to mention that the symposium features top-notch female researchers entirely, 3 of which of us are early career and 2 of which are new to ISDP. So I have recruited them into ISDP. Please give them a warm welcome. And so we were so honored to be chosen as the presidential symposium. This is a topic that's been near and dear to my heart for a very long time as I transitioned from nutrition research interventions to stress physiology. And really the question of how do developing stress response systems interact with physiological processes related to nutrition? And how does this interaction change or deepen what we already know about development? Just broadly, but also can they elucidate new pathways to mental and physical health and well-being? And so in this symposium, we'll begin with a rodent model that spans prenatally into adulthood with Dr. Mariann Paulsen, who will present her work exploring sex-specific links between a translational model of prenatal maternal overnutrition and offspring trajectories of hypothalamic-glucocorticoid receptor signaling. Dr. Paulsen is an assistant professor in the Department of Pediatrics at the University of Minnesota and a faculty member of neonatology. Next, Mariann Howland will show in a sample of humans how maternal pregnancy HPA-axis activity relates to gestational weight gain and obesity risk among women and their children. Mariann will be joining us virtually from the University of Minnesota, where she is a doctoral student there with me and Gunnar at the Institute of Child Development. And then I have the immense privilege to present my work as a postdoctoral fellow in the Department of Psychiatry and Human Behavior at Brown University, where I'm working with Dr. Laura Stroud. And my presentation will echo and extend Dr. Paulsen's work in humans, examining how financial adversity or a chronic stressor interacts with maternal overnutrition to predict newborn cortisol reactivity. Our 4th presentation is from Dr. Patricia Silveira, who will be joining us remotely from McGill University. And she will directly connect preclinical animal models, a very exciting method that she's really pioneered, and human cohort studies with research that draws on rodent model gene networks to predict risk for cardiometabolic and psychopathology comorbidities in children and adults after exposure to adversity. Finally, Dr. Gunnar will serve as a discussant of this symposium. And at this point, I think she needs no introduction, but I'm looking forward to her always entertaining and incisive discussion on the interaction of nutrition and stress-sensitive systems with all of us. So we'll end our symposium with a question and answer session with all of you. And so please join me in welcoming our first speaker, Dr. Mariann Paulsen.
I would like to thank ISDP, Drs. Reid and Gunnar, for the opportunity to present my research here today. I have no disclosures to report. In 2014, 74% of adult men and 67% of adult women were overweight and obese. While the prevalence of adults who are overweight has been steady over time, the prevalence of obesity has significantly increased among adults. Obesity is higher in women compared to men. 40% of women and 35% of men have obesity in adulthood. Extreme obesity is nearly twice as prevalent in women compared to men. 10% of women compared to 6% of men have extreme obesity. And more recently, the prevalence of obesity has increased significantly among women, but not men. 2/3 of women enter reproductive age overweight or obese, making this chronic condition the most common complication of pregnancy. Currently, women who have obesity do not have additional recommended metabolic screening during pregnancy outside of an early glucose tolerance test. A large volume of epidemiologic evidence has demonstrated the strong association between maternal nutrition and development of obesity in offspring. The maternal obesity, hormonal milieu, and nutrient excess mediate alterations in fetal organ development that cause obesity risk throughout the life cycle. Currently, there are also no routine perinatal biomarkers used to identify obesity risk in offspring born to mothers with obesity. And this may be in part because the mechanisms which cause maternal offspring obesity transmission have not yet been identified. A study following a Midwestern cohort longitudinally demonstrated a stronger relationship between mother and daughter BMI compared to mother and son BMI. In the figure on your left, the y-axis shows adjusted standardized beta coefficients for the relationship between mother and child BMI. The x-axis is child age from birth to 18 years. The line with triangle symbols, also the top of the figure, represents the relationship between mother and daughter BMI. The line with circle symbols, also the bottom line of the figure, represents the relationship between mother and son BMI. Mother and daughter BMI correlations were both significant earlier in life and were stronger compared to mother-son BMI correlations. This is all to demonstrate that collective previous work tells a story of dangerous cycle of transgenerational transmission of obesity, as well as a critical need to understand metabolic mechanisms causing obesity transmission.
But that's because you're noted as a panelist, but others.
Our lab investigates the hypothalamus as a key mediator in the development of offspring adiposity following exposure to maternal obesity. Our and my enthusiasm for the hypothalamus is founded on four key points. First, the hypothalamus is the master regulator of metabolism and energy homeostasis.
I do.
Defects in hypothalamic development cause obesity.
I didn't have it connected on this.
Second, the hypothalamus has the most complex circuitry of any brain region as it integrates both sensory information about the internal environment and directs actions to restore homeostasis through autonomic neurons and neuroendocrine signals. Additionally, the hypothalamus is unique to all other regions, unique to all other brain regions. In addition, it connects the peripheral organs such as adipose tissue. Next, the hypothalamus is exquisitely sensitive to injury during its development. The majority of neuron migration, axon extension, dendritic aberration, synaptogenesis, and circuit remodeling elaboration take place during the 3rd trimester of pregnancy. Last, the developing brain, especially the hypothalamus, is an incredibly resilient organ. Its plasticity has been reported throughout the lifetime, but most notably during development. Therefore, if modifiable mechanisms causing obesity development can be identified, this period of neuroplasticity can be leveraged to improve health outcomes in infants born to mothers with obesity. In particular, my lab is interested in maladaptive hypothalamic programming of the glucocorticoid receptor as a potential critical mechanistic link to explain the association between maternal and offspring obesity. The glucocorticoid receptor is expressed throughout the hypothalamus. Glucocorticoid signaling and regulation are paramount to brain development and lifelong metabolic regulation and energy homeostasis. Transgenic and loss of function preclinical models leading to decreased hypothalamic glucocorticoid receptor expression causes HPA-axis hyperactivity, hypothalamic growth impairment, decreased activity, and peripheral metabolic syndrome in adulthood. Additionally, females are more sensitive to HPA-axis programming compared to male counterparts when exposed to maternal stress or trauma during development. If maternal obesity and overnutrition are similar types of stressors to the fetus, as demonstrated in previous research, much of your research, Altered glucocorticoid receptor signaling may be a potential mechanism contributing to the increased prevalence of obesity in women compared to men. Our overarching hypothesis is that the association between maternal and offspring obesity is caused by maladaptive hypothalamic programming following intrauterine exposure to maternal obesity. We are particularly interested in studying male and female offspring separately, as women who develop obesity will re-enter the cycle during reproductive age, thus contributing to the transgenerational cycle of obesity. We hypothesize that the hypothalamic phenotype, and specifically glucocorticoid expression, will be more severe in female offspring compared to male offspring exposed to maternal obesity. To test this hypothesis, we use an established and clinically relevant mouse model of mother-offspring obesity. I will summarize this that we feed mom dams Big Mac Coca-Cola sort of diet, and they give birth to the pups, and we challenge them later in life with either just a regular diet, a standard chow diet, a high-fat, high-sugar diet, or we challenge the females with pregnancy. And the important point here to know is that pups, that when they are born, their hypothalamus is fully developed towards the end of lactation, which is 2 to 3 weeks of life, whereas in humans, it's at time of birth. There we go. This is a metabolic profile of the dams towards the end of gestation, early lactation period, and it essentially shows that they are non-diabetic. They pass their glucose tolerance test, but they are insulin resistant, and they have an abnormal adipokine profile. All right, I think we're caught up now. We are interested in investigating glucocorticoid programming caused by maternal obesity. So we measured plasma corticosterone, that's the active glucocorticoid hormone in rodents, opposed to cortisol in humans, as well as plasma cholesterol, which is the rate-limiting step for fetal cortisol synthesis. Pregnant dams fed an obesogenic diet have 1.8-fold higher corticosterone and 30% higher cholesterol compared to control dams. We speculate that higher maternal cortisol and or cholesterol causes higher cortisol exposure and cortisol synthesis respectively in the developing fetus. Male and female offspring born to obese mothers have a similar adiposity phenotype in adulthood. We use quantitative MRI and cortisol-sensitive abdominal fat pad weight to quantify adiposity in offspring. Male and female adult offspring exposed to maternal obesity and fed a standard postnatal chow diet had significantly higher adiposity. Female offspring compared to male offspring had 23% higher adiposity, suggestive of sex-specific mechanisms contributing to adiposity despite a similar metabolic phenotype. Adult offspring exposed to maternal obesity fed a postnatal high-fat, high-sugar diet had 30% higher percent body fat compared to adult offspring exposed to control dams. And then in this last figure, we exposed female offspring to control and high-fat, high-sugar diet during pregnancy, and then measured abdominal fat pad at the end of gestation. And we found that pregnant female offspring exposed to maternal obesity had 2-fold higher abdominal adiposity compared to control. Male and female offspring born to obese mothers have abnormal energy balance in adulthood. Using metabolic chambers, we measure food intake, basal metabolic rate, and activity in adult offspring. Female offspring exposed to maternal obesogenic diet had 19% higher food intake, 11% lower basal metabolic rate, and 30% lower activity compared to female offspring exposed to maternal control diet. Male offspring exposed to a maternal obesogenic diet had 10% lower basal metabolic rate compared to male offspring exposed to a control diet, and male offspring groups did not differ in food intake or activity. Female offspring in general had 40% higher activity compared to males. The hypothalamic-melanocortin system is the rate-limiting step in total body energy homeostasis. Male and female offspring born to mothers on obesogenic diet had 33% lower hypothalamic melanocortin-4 receptor gene transcription compared to offspring born to mothers on control diet. Lower melanocortin-4 receptor expression is associated with obesity and metabolic disease, representative of an imbalance between energy in and energy out. As glucocorticoid signaling plays a significant role in brain development, we are interested in its programming role in offspring exposed to a maternal obesogenic diet. We measured glucocorticoid receptor, or NR3C1, transcript expression at fetal, peripubertal, and adult time points. We found that female, but not male, offspring born to obese mothers had decreased hypothalamic glucocorticoid receptor transcript expression during fetal life, early adolescence, and adulthood. At embryonic day 18.5, female offspring born to obese mothers had 57% lower hypothalamic-glucocorticoid receptor transcript expression. At 5 weeks of age, 33% lower expression, and adulthood, 54% lower expression compared to offspring exposed to maternal control diet. Because the glucocorticoid receptor is a key regulator of the hypothalamic-pituitary-adrenal axis, we measured hypothalamic corticotropin-releasing hormone, or CRH gene transcript, plasma adrenocorticotropin hormone, or ACTH, and plasma corticosterone longitudinally. Our adult studies have not been completed yet. However, our assessment at 5 weeks of age, a correlate to peripuberty in humans, demonstrates 26% higher hypothalamic CRH transcript expression, 39% lower plasma ACTH, and 49% lower plasma corticosterone in female but not male offspring exposed to a maternal obesogenic diet compared to offspring exposed to a maternal control diet. There is an anticipated stress hyporesponsive period characterized by high CRH and low ACTH and cortisol. In rodents, this expected stress hyporesponsive period occurs postnatally during the first 2 weeks of life. Both preclinical and clinical studies have suggested that this occurs in maternal stressors such as trauma. However, this has not been shown in nutritional-based research before. To gain insight into what maternal mechanisms will be driving HPA-axis programming, we performed correlational analyses of our maternal offspring cohorts. These analyses are preliminary and limited by very small sample sizes. We found significant linear relationships between maternal corticosterone and adult female hypothalamic glucocorticoid receptor expression. Additionally, we found significant linear relationships between adult female offspring hypothalamic CRH and maternal cholesterol, maternal corticosterone, maternal ACTH, and maternal insulin. Further translational studies are necessary to confirm these relationships. This preliminary data, however, aligns with our hypothesis that the hypothalamic glucocorticoid receptor programming may be a critical mechanistic link in the association between maternal and offspring obesity, as well as its transgenerational transmission from mother to daughter. In summary, offspring exposed to maternal obesogenic diet have increased adiposity and altered hypothalamic function characterized by energy surplus. Adult offspring exposed to a maternal obesogenic diet had decreased basal metabolic rate and hypothalamic melanocortin-4 receptor, which is associated with decreased energy output from the body. Additionally, female offspring exposed to maternal obesogenic diet demonstrated higher appetite or energy intake and decreased activity or less energy out. Collectively, these findings suggest maternal obesogenic diet causes an obese phenotype in offspring mediated by the hypothalamus. We speculate that increased adiposity in female offspring exposed to maternal obesogenic diet is caused by abnormal glucocorticoid receptor expression and HPA-axis feedback and or response. We speculate that females are at an even further risk of increased adiposity when exposed to postnatal stressors such as an obesogenic diet, trauma, chronic stress, or pregnancy. Therefore, the hypothalamus and its role in the HPA-axis may be a critical mechanism involved in the epidemiologic differences found in higher female prevalence of obesity compared to males. I would like to thank Dee Kohonik, who did perform much of this work presented here today, and I'd like to thank my collaborators and those who've supported my research. I'd especially like to thank my mentors, Dr. Gunnar, Dr. Rao, Dr. Bartolomucci, and Dr. Sequest, and thank you for the opportunity to present my research today and for your attention.
All right, so now we're going to go to some pre-recorded presentations if I can figure out how to do that. Is it showing up on the Zoom? Yep, okay, great.
Hi, my name is Mariann Howland. I'm a fourth-year PhD student in developmental and clinical psychology in the Institute of Child Development at the University of Minnesota, working with me and Gunnar. I'm really grateful to Brie for putting together this symposium. I'm really excited to be a part of it. Sad that I'm not there in person, but appreciate the opportunity to participate virtually. I'm going to be presenting findings from my ongoing collaboration with my postdoc mentor, Dr. Laura Glenn, who's a professor at Chapman University and a project director within the UC Irvine County Center. These data that I'll be talking about are drawn from the center project, which I worked on before coming to grad school. So 30 years ago now, the epidemiologist David Barker put forth his seminal hypothesis on the fetal origins of adult disease. And in the years since, evidence has really accumulated suggesting that stress and nutritional exposures in fetal life program physiological set points, increasing risk for physical health outcomes like obesity and cardiometabolic diseases later in life. Stress and nutrition during pregnancy are linked with obesity all the way into adulthood and also are associated with more proximal indicators of obesity risk, such as birth weight and postnatal growth. And while less often considered, gestational physiological processes also are linked to long-term maternal phenotypes. The physiological demands of pregnancy-induced transient metabolic syndrome are proposed to act as a stress test for maternal body systems, which can uncover maternal risk for later disease. For example, women who develop gestational diabetes are at higher risk for obesity and cardiometabolic disease a decade later. One pregnancy outcome that is shown to confer risk for later life obesity and cardiometabolic disease in both mothers and their children is excessive weight gain over gestation, and this is particularly the case for women who enter pregnancy with a high BMI. Recent U.S. population level data highlight that excessive gestational weight gain is a significant public health concern. The graph on the left of the slide here shows the proportion of women with adequate in green, inadequate in blue, and excessive in red gestational weight gain, with the overall sample rates shown in the leftmost bar. Overall, only about 1/3 of women gaining the recommended amount of weight during pregnancy, with almost half gaining weight above the recommended amount for their pre-pregnancy body mass index. This rate increases to more than half among women with pre-pregnancy BMIs in the overweight or obese range, shown in the rightmost 4 bars in this graph. Today, I'm going to be focusing particularly on the relationship between excessive gestational weight gain and offspring obesity risk, with previous studies linking excessive gain with risk beginning in childhood and into adulthood, as detailed in a 2014 meta-analysis by Mariann and colleagues. While there's good evidence to suggest that maternal body composition and excessive gestational weight gain have compounding effects on offspring obesity risk, the biological pathways underlying these associations are not well understood at this point. In the larger fetal origins literature, gestational stress physiology is a primary proposed mechanism of programming. Maternal hypothalamic-pituitary-adrenocortical HPA-axis activity during pregnancy may link gestational weight gain with proximal indicators of obesity risk, such as higher infant birth weight, and also with later life outcomes. Pregnancy involves substantial changes in maternal HPA-axis function, which are largely a result of placental production of corticotropic-releasing hormone, the master regulator of the HPA-axis. Placental CRH is released into the maternal circulation, where it stimulates maternal HPA-axis activity. And maternal and fetal cortisol actually upregulate rather than inhibit placental CRH production, and this positive feedback loop normatively results in progressive rises in maternal cortisol over gestation, with maternal cortisol levels reaching levels seen in Cushing's disease by the end of pregnancy. And I'm showing here on the right-hand side of this slide levels of CRH, ACTH, and plasma cortisol in the maternal compartment over gestation. And while placental production of the enzyme 11-beta-HSC2 offers the fetus some protection from exposure to maternal cortisol, it is only a partial barrier, with an estimated 40% of the variance shared between maternal and fetal cortisol levels during mid to late gestation. Elevated fetal exposure to maternal cortisol late in gestation benefits the fetus by promoting growth and organ maturation. Increasing evidence highlights the importance of gestational timing when considering associations between cortisol exposures and child outcomes. Exposure to elevated cortisol early in gestation is shown to have consequences for fetal development and later physical and mental health outcomes, while elevated cortisol levels late in gestation are linked with positive outcomes consistent with the benefits of cortisol for fetal organ maturation late in pregnancy. The graph on the left of this slide is from a recent study from Laura's group, showing trajectories of maternal plasma cortisol over gestation identified by growth mixture modeling. Women demonstrating a flat profile of cortisol output over gestation, specifically a lack of an increase in the latter half of gestation, represented by this green trajectory line, reported the highest levels of psychological distress over gestation. Lower levels of cortisol late in gestation are also associated with less optimal neurological and cognitive development from the fetal period into childhood, and also with child cortical thickness, less cortical thickness. This pattern of salutary effects of elevated cortisol in late gestation appears to persist until lactation, during which lower cortisol in breast milk is associated with greater BMI percentile gains among female infants across the first 2 years of life, the trajectory highlighted in bold here on the right graph, and lower breast milk cortisol is linked with higher BMI percentiles in both sexes by 24 months of age, reflecting increased risk for later obesity. Interestingly, in this sample, higher gestational weight gain was associated with lower breast milk cortisol. So in the current study, we wanted to examine whether this gestational weight gain and lower cortisol linkage is apparent beginning in pregnancy, and whether lower cortisol levels late in gestation also are associated with a proximal indicator of obesity risk in the birth weight, which would suggest it may be a programming mechanism linking gestational weight gain with obesity risk. So to address this question, in a separate longitudinal cohort of women and full-term infants than the cohort I was talking about in the previous slide, we examined associations between gestational weight gain, pregnancy, maternal hair cortisol, and infant birth weight. Given evidence that women with a high pre-pregnancy BMI are at higher risk for gestational weight gain and also appear to be more vulnerable to the effects of excessive gestational weight gain, we stratified our analyses to consider whether associations differed among the 60 women in the subsample with a pre-pregnancy BMI in the normal range versus the 66 women with a high BMI in the overweight or obese range. Pregnant women in this sample were recruited during their first trimester from prenatal clinics and from the university community in Orange County between Southern California between 2013 to 2017. Sample characteristics are displayed here. Women were on average 29 years old. 36% of women had a bachelor's degree or higher, and the majority were cohabitating with the baby's father. The median annual household income was approximately $50,000, and just for reference, the median household income for the county in 2019 was $90,000. Roughly half the sample identified as Hispanic Latina, and another third identified as white or Middle Eastern North African. 12% of women had a diabetes diagnosis and 12% a hypertension diagnosis in the pregnancy. Only 2 of these women had both types of diagnoses. All infants in the sample were full term, as I said, with a mean gestational age at birth of 39 weeks, and 44% of the infants were female. Maternal gestational weight gain was simply the difference between pre-pregnancy weight and weight at delivery. Here I indicate the percentage of women in our sample in the normal and high pre-pregnancy BMI groups with excessive, adequate, or inadequate weight gain over gestation, according to the 2009 Institute of Medicine weight gain recommendations. As shown in the red shaded portion of the bars, 56% of women with a high BMI showed excessive weight gain over gestation as compared to 33% of women in the normal BMI range. Infant birth weight percentile was derived using World Health Organization growth standards, and the 2 maternal BMI groups did not differ in average birth weight percentile. Early and late gestation maternal cortisol was measured in 3-centimeter hair samples collected at approximately 15 and 35 weeks gestation, reflecting cortisol production from approximately 2 to 15 weeks gestation, and then from approximately 22 to 35 weeks gestation. Hair samples were assayed for cortisol by liquid chromatography and tandem mass spectrometry. And as expected within individual hair cortisol levels were higher in late gestation compared to early gestation. Correlations and regressions were conducted separately by group. So on the following slides, I'm going to share graphs depicting bivariate associations, but just for reference, these findings persisted when adjusting for covariates, which were household income, cohabitation with the baby's father, and diabetes or hypertension status in the pregnancy. So first, I'm going to show that in both groups, greater gestational weight gain on the x-axis here was associated with higher infant birth weight percentile on the y-axis. Next, I'm going to unpack associations between gestational weight gain and hair cortisol. This graph shows the 2 gestational time points on the x-axis and log transformed hair cortisol on the y-axis. The gold lines on this graph are women with high pre-pregnancy BMI, and the gray lines, which are essentially perfectly overlapping, are women with normal BMIs. Gestational weight gain was modeled continuously in these analyses, but for illustrative purposes on this slide, I'm showing solid lines as representing women with higher gestational weight gain, So 1 standard deviation above the overall sample mean, and then the dashed lines are women with lower gestational weight gain, so 1 standard deviation below the overall sample mean. So first you can see that women with high pre-pregnancy BMIs, the 2 gold colored lines, on average had higher levels of paracortisol compared to women with normal range BMIs, both in early and late pregnancy. There were no associations between early pregnancy paracortisol and gestational weight gain in either group. And then among women with a high BMI, greater gestational weight gain was associated with lower late pregnancy paracortisol, where there was no association between late pregnancy paracortisol and gestational weight gain in the normal range BMI group. So here's the late gestation finding shown continuously. So again, showing that greater gestational weight gain was associated with lower late pregnancy paracortisol levels only among women with high pre-pregnancy BMIs in the gold, showing the gold line. And then lastly, among women with high BMI, lower late pregnancy paracortisol was associated with higher infant birth weight, whereas there was no association among the normal range BMI group. And if anything, the pattern is in the opposite direction. So to summarize the findings for both groups, we saw that greater gestational weight gain was associated with higher infant birth weight, a proximal indicator of obesity risk. The magnitude of this association was stronger among women with a high pre-pregnancy BMI, consistent with the heightened risk for this group. Women with higher pre-pregnancy BMIs had higher levels of paracortisol during both early and late pregnancy as compared to women with BMIs in the normal range, and this is consistent with previous evidence of these group differences, including data that Megan just shared. And then only among women with a high pre-pregnancy BMI do we see that greater gestational weight gain was associated with lower late pregnancy paracortisol, which in turn was associated with higher infant birth weight percentiles. And these associations were not evident among women with BMIs in the normal range. These findings suggest that this possible biological pathway may be specific to women who enter pregnancy with a high BMI. We interpret these findings really in light of the established salutary effects of heightened cortisol late in gestation for fetal organ maturation. These findings speak more broadly to the importance of considering the timing of prenatal cortisol exposures when examining their potential programming effects. And our findings linking greater gestational weight gain with lower late pregnancy hair cortisol levels and lower cortisol levels then being associated with higher infant birth weight percentiles are consistent with data from a previous study from our group in a separate cohort of women and infants in which greater gestational weight gain was associated with lower cortisol and breast milk, which in turn was associated with higher BMI percentiles at 2 years of age. Primary limitations of this study are a modest sample size and the fact that we did not look at trajectories of weight gain over gestation, nor did we look at postpartum patterns of child growth or maternal weight loss or retention. Assessment of postpartum maternal and child weight trajectories is an important future direction, as would be an attempt to directly replicate previous findings linking lower breast milk cortisol to child obesity risk. It also would be interesting to assess HPA-axis activity in these children to see if the lower cortisol production among high BMI women with greater gestational weight gain would also be evident in their children, as Megan's rodent findings suggest may be the case. More broadly, these findings suggest that maternal HPA-axis activity is one of likely multiple pathways by which gestational weight gain engenders in utero adaptations, which program the set points of fetal organ systems to shape long-term obesity risk in offspring, and possibly also women's own risk for later obesity and cardiometabolic diseases. These results and the broader symposium highlight that more research should focus on understanding the interplay between gestational stress and nutrition in shaping maternal and child physical health trajectories. And finally, gestational weight gain is clearly a significant public health challenge and should be a target for interventions to support healthy maternal and child development. So I want to thank Brie again for organizing this symposium. I want to thank Megan for serving as our discussant and in her role as my doctoral advisor for always supporting my independent research interests and collaborations. I want to thank my co-author on this project, Dr. Lori Glynn, for the opportunity to work with this data and her continued mentorship and support. I want to thank my mentors and the PIs of the larger NIMH Conti Center at UCI, which funded this study, especially Dr. Kurt Sandman and Alicia Davis. I'm funded by an NSF GRFP, and then I lastly want to thank ISDP for giving us the opportunity to present this symposium. Thanks so much for listening, and I look forward to our discussion.
It looks like we have been signed out again from Zoom, so I don't.
We still have you on camera here.
Is it still going? Okay. Great, because we've been completely signed out, so I don't know. I hope that doesn't mean anything. You can still see it on, okay, cool. I'll take it. All right, you can still see that? Okay, great. I'm not, I guess we're still here, so, okay. Perfect. So thank you so much. For those of you who are online and might not see the rest of my body, the irony of presenting on this while 7 months pregnant is not lost on me. So yeah, we'll just roll with it. But I'm really excited to be talking to you all about prenatal material adversity, BMI, and newborn cortisol reactivity. And you can find me on Twitter at Brie Reid. So just a shout out to the ISDP social media team in advance. If I can, oh, there we go. So as has been discussed through the last few presentations, pregnancy is really considered the sensitive period for both stress and nutrition exposures. One reason is that human brain development really lays this healthy foundation to build the rest of the house for the brain architecture across life in the first 1,000 days. And so my conceptual framework that I really work within is the intersection of early stress and poor nutrition, both from conception and infancy and early childhood, how they get embedded under the skin, how they become biologically embedded through physiological disruption to developing systems, and how that leads to poor mental and physical health outcomes. So that's kind of where I'm coming from. And so the system of focus, as it has been and as it will be for the next couple of slides or next presentation, is the hypothalamic-pituitary-adrenocortical axis. It's one arm of the mammalian stress response system. And we just love cortisol because we can measure it in saliva for humans and plasma as well for rodents. And do you guys measure it anywhere else? The tissue. In the tissue, I mean, they just get to take all kinds of fun samples, but we focus really on hair and saliva, and it really does play, as far as we know, a role in early development and targets the brain and a lot of aspects of the body as well. So I'll be talking about two components of the stress response system. One is the diurnal cortisol rhythm, which is characterized by this peak within the first 30 to 45 minutes after wake up. And then it kind of, you can see that it reaches this low point ideally at bedtime. And then the cortisol reactivity to stress, which we have talked about a little bit, but really in an ideal system, when we experience a stressor, like the Zoom recording going off during the presidential symposium, about 20 minutes later, so probably right where I'm at, you want to see this rise in cortisol in saliva, and then this nice little drop as we recover. So it's all about the flexibility. As Mariann talked about, the HPA-axis activity is dramatically altered in pregnancy. And the diurnal cortisol rhythm, the cortisol awakening response, so that peak right at wake up, decreases across gestation. So there is this kind of decline of the cortisol awakening response, or the CAR. Changes in typical patterns of HPA activity have been associated with poor neonatal outcomes and infant temperament. And pregnancy CAR significantly, in a recent study with 9-month-old infants, did significantly predict both infant cortisol and behavioral responses to a repeat separation stressor. So dysregulated maternal cortisol is one plausible mechanism that links maternal exposures to nutrition and stress to offspring outcomes. So the HPA-axis in newborns is there. I will say the stress response is present, and it really peaks at around 2 months from earlier research. And there's a thought that a dysregulated response might carry implications for the programming of both the infants' HPA-axis and also behavioral outcomes across development. So it's kind of a Goldilocks and the three bears situation. Both a hyper response and a hypo or blunted response is considered unideal. Although how you define that in any given sample can be a challenge. So we'll just kind of put a pin in that for later. But there have been a few studies that look at these systems. So infants of mothers who exhibit elevated cortisol levels in pregnancy have been shown to have high cortisol levels after birth. And then there's a thought that neonates who are more reactive with the stress response system may carry a greater risk of different kinds of developmental outcomes, such as behavioral inhibition, maybe increased stress reactivity across development, poor attention, emotion regulation. And so my conceptual framework for this talk is really shifting our focus, as I know a lot of us are stress researchers. And so a lot of us operate in this, we assume the nutritional environment is fine or the same between our stressed and non-stressed groups. And we just are looking at stress as the exposure of interest. And in contrast, my past life as the nutrition intervention researcher, they really just think about stress being the exposure and target of intervention, and really or nutrition being the target of intervention. And if they talk about stress at all, we're really lucky, but it's not really considered, although both of these systems interact physiologically. And just like glucocorticoids have the word, the root gluc in it, there's a glucose metabolism that's involved in the system inherently. So I'm really interested in this interaction, how they might differentially impact offspring cortisol reactivity and subsequent outcomes. So we're going to use some imperfect measures to characterize this. One is a proxy for chronic stress. And so we're looking at economic hardship or material adversity, or really income here in pregnancy, which has been associated with both worse health status and elevated cortisol across the day. And then for our metric of nutrition, we're looking at pre-pregnancy BMI and gestational weight gain as sort of our imperfect proxy of the nutritional environment. So I don't want to judge this stock photo of the woman with the donut and coffee, because that was like me 8 hours ago. So it's just what we have. So we need more stock photos of women in pregnancy, I guess. I will be looking at a subset from the Bambi study with Dr. Laura Stroud. And so this was an R01 that was conducted between 2007 and 2011. And so we're looking at a subsample of 104 with data longitudinally on both pregnancy and offspring cortisol, pre-pregnancy BMI, and offspring head circumference as kind of a metric of health at birth. For the following analyses. And so if we look at the timeline to our right, we get our demographics, so our stress and so our financial adversity, our stress exposures, as well as our pre-pregnancy BMI at intake before 20 weeks gestational age, around 16 weeks gestational age, so pretty early. And then at 35 weeks in pregnancy, we look at maternal cortisol across the day, and that was done with 3 days, 3 samples, wake, 30 minutes after wake, and then our bedtime levels. We looked at gestational weight gain from pre-pregnancy BMI to 35 weeks. And then at delivery, we had our infant health and demographic information. And then we did an infant cortisol kind of stressor, or the neonatal neurobehavioral, or ENDS exam, which I'll get into, to collect infant cortisol reactivity. And so just to give you an idea, I will put a shameless plug for my own daughter in that baseline photo, because if you have a baby, why not plaster them all over your slides? But what we do is we bring them into the lab both within the first week of life, and we'll be looking at the first about a month post-delivery. So we bring them into the lab, we get a baseline saliva sample, and then we conduct this ENDS exam, which is if anyone has been to a pediatrician with a newborn, you do a lot of manipulations, and then there's also some stimuli and attentional processes that the infants have to do. It has reliably provoked a stress response in Laura Stroud's cohorts, and we have 2 we have 3 other samples of cortisol at the end of the ends, because sometimes it's so stressful that we have to end the exam early before we get that 20-minute sample. Then we get it at 20 minutes and then at 40 minutes, because so different parts of the exam can stress different infants. There's sort of individual differences there in terms of which ones provoke this stress response with salivary cortisol. So just to give you kind of an overview of the sample, we have a fairly diverse racial ethnic breakdown in this cohort of mothers who were recruited primarily from Rhode Island and the surrounding areas. The income also, you can see, is pretty spread out in terms of the range from under $10,000 per year to over $100,000 per year. So we feel good about that spread. And then mean maternal age was around 27. Mean pre-pregnancy BMI was a little on the, kind of on the cusp of normal to overweight with the BMI. And gestational age at birth was 39.5 weeks, which is always what we like to see. And 44% of the offspring were female, which is scarily the same number that Mariann's population was somehow. So for the analyses, we looked at a mediation model between the stress and nutrition exposures, maternal cortisol awakening response at 35 weeks, and then this newborn cortisol reactivity at 1 month old. And so for cortisol reactivity for the newborn, we looked at baseline to peak cortisol across the ends. Interestingly, pre-pregnancy BMI and income predicted maternal cortisol awakening response, but then the interaction of the two did not. And they also did not predict newborn cortisol response, but the interaction did. So we can kind of break that into it. And maternal cortisol awakening response also was associated with newborn cortisol response, but there was no mediation between these nutrition exposures through maternal cortisol to infant cortisol, which was surprising, but cortisol never does what we want it to do. So returning to our question of how might the interaction of prenatal maternal stress and nutrition differentially impact offspring cortisol reactivity, this is just a rough graph of the interaction here with the lowest income in red and the highest income bracket in blue. And so mothers with the highest pre-pregnancy BMI and the lowest income, so kind of this dual hit, were more likely to have offspring with higher cortisol responses to the ends or the neurobehavioral exam. And I just highlighted in yellow, 20 to 30 is about the normal range of BMI, or normal to just overweight, not obese. So you can see that a lot of the action is really happening at the tail ends of the BMI. So always something that's interesting. So pre-pregnancy BMI was associated with increased cortisol awakening response. There have been links between maternal obesity and elevated cortisol release, so that might be playing a role. It's also possible that obesity and pre-pregnancy BMI is really capturing a lot of other factors. So either biological factors such as disrupted sleep, increased inflammation, or medical comorbidities, or some social factors such as stigma, weight-based stigma, or self-esteem and body image, which all have been associated with cortisol in non-pregnant populations. Gestational weight gain, I didn't really talk about this in the results, but I feel like given our previous presentation, I should address it. It did predict maternal cort, but it did not predict infant cortisol. So it might just be that there was an interaction there that we are just not powered to detect with gestational weight gain interacting with actual pre-pregnancy BMI. Higher income was also associated with lower maternal cortisol awakening response, which does kind of follow in line with both non-pregnant samples in some contexts and also a sample of pregnant and ethnically diverse low SES women. But again, CAR can kind of do different things with different income levels depending on the sample you're looking at. So just something to think about. Higher maternal CAR was associated with lower infant cortisol reactivity. And so this was similar to the study that was previously discussed in 9-month-olds with the lower CAR associated with decreasing infant cortisol response, and higher CAR actually was related to more stable cortisol response and more stable behavioral outcomes during repeat separations. Again, that's at 9 months, so take that with a grain of developmental salt. And then there were no indirect effects from predictors of maternal CAR to infant cortisol, so they seem to be doing their own thing. So maternal nutrition and maternal adversity exposures may influence offspring cortisol reactivity independent of maternal cortisol late in pregnancy. Some things to keep in mind: pregnancy is complicated and the endocrine system is frustratingly complicated. And so the maternal endocrine context reflects the maternal context, but also there's all kinds of things obviously happening within the fetal compartment. Placental CRH is released both into the maternal and fetal compartment. So even though the infant and mother cortisol outcomes kind of did their own things, depending on the exposures, that might be reflective of kind of different, I guess, context sensitivities. And of course, Laura Stroud's work has this additional complexity of the placenta glucocorticoid and serotonergic pathways that also differentially impact offspring COHORT. So looking ahead, we plan to look at dual exposures and cortisol trajectories across pregnancy to see if these dual exposures predict different trajectories across pregnancy in a larger cohort of around 400. And then we also just submitted a grant, so a little teaser and a prayer to the NIH gods to look at how chronic stress and HPA-axis patterns across pregnancy may directly impact nutrient status in both mother and offspring, specifically with iron deficiency and hepcidin regulation. So with that, I want to thank you, both the audience here and also Laura Stroud in the Bam Bam Lab and the amazing T32 that I'm on at Brown University. So thank you so much. Okay, and now I have the pleasure of navigating the second to last. All right.
Good afternoon. Thank you so much for the invitation to participate on this symposium. It is a delight to have the opportunity to talk about the work we have been doing in my lab and be presenting alongside these talented colleagues. When I launched my lab, we focused on the effects of being born small for gestational age on child behavior and risk for disease later in life. In a series of studies in human samples and rodents, we showed that poor fetal growth is linked to increased preference and impulsivity towards palatable foods such as sugars and fats at different ages during the life course. This altered behavioral phenotype is associated with differences in the neurobiological responses to palatable food images in our neuroimaging studies, with increased activation of areas related to impulse control. We believe that these effects and the subtle differences in eating behavior throughout the life course contribute to the well-known increased risk for metabolic disease later in life in these individuals born as children. These individuals are known for these increased risk for type 2 diabetes, cardiovascular disease, as well as their psychiatric comorbidities like depression, ADHD, anxiety, and addiction. To better explore the mechanisms involved in the relationship between being born small for gestational age and altered behavior, we standardized an animal model of poor fetal growth based on 50% caloric restriction from the daily control food intake from pregnancy day 10 onwards. In this model at birth, all pups are adopted by control dams so that the effect of the undernutrition is located only during the fetal period. It is interesting to note that this model recapitulates the classical human tryptic phenotype, as pups are born as GA but catch up on growth before weaning and have hyperinsulinemia in adulthood. We were able to see the same increased preference for palatable foods that we had observed in the human cohorts in comparison to control animals. And in a delayed gratification test, in which the animals were trained to choose between a small immediate reward or a delayed or a large reward, we saw that animals born as GA in most instances cannot wait for the large reward, showing a behavioral pattern of impulsivity towards palatable foods that is very similar to what we have observed in children born as GA at 3 years of age using the SNAP delay test. Based on that, we can design a framework in which early life adversity reflected in poor fetal growth leads to an altered behavioral phenotype of increased preference and impulsivity towards palatable foods, altered sensitivity to reward, and altered cognition. Now, thinking about the possible neurobiological mechanisms associated with alterations in these phenotypes, we hypothesized that changes in the cholinergic signaling could be involved. So, using chronoperometry, we were able to measure dopamine release in the mesocortical limbic system in awake animals as they encountered and ate palatable foods. So, during behavioral performance. And we saw that intermediate prefrontal cortex, adult animals born as GA had decreased dopamine release when facing the palatable food stimulus. And in the nucleus accumbens, adult animals born as GA had longer time to reach the dopamine peak when facing palatable food specifically, as there were no differences in response to regular and standard chow. And interestingly, these differences in the time to reach dopamine peak in the nucleus accumbens were completely reversed by a previous injection of insulin, suggesting that the modulation of dopamine neurotransmission by insulin is a possible mechanism by which prenatal adversity modifies the behavioral phenotype and the risk for disease in the long term. We recently performed whole genome RNA sequencing in Israeli samples of newborn pups that were exposed to the SGA model. We extracted RNA and submitted for sequence. And after processing the data, we used the raw counts for each gene in all the animals in a multivariate analysis called weighted gene co-expression or WGCNA analysis that serves to identify gene co-expression networks. We did identify several modules of co-expressed genes, but for now, let's look only at the brown module that is significantly associated with the exposure to food restriction during gestation. It has more than 900 genes that form this network that we see here. And interestingly, when performing the enrichment analysis of the pathways and gene ethology processes associated with this network, we see significant processes like the role of stress on obesity and type 2 diabetes, inflammation, development of the dopamine neurotransmission, and response to insulin in accordance to the findings that I showed you before. Then we can complete our framework, again, in which early adversity leads to altered behavior phenotype, and it seems that variation in the cholinergic neurotransmission in different regions of the mesocortical limbic system of the brain, like the nucleus accumbens and the prefrontal cortex, seems to be a mechanism that explains the behavior phenotype and that likely contributes to the development of cardiometabolic and psychiatric comorbidities in the long term. Moreover, we also show that at the neurochemical level, There's differential modulation of the dopaminergic signaling by insulin in fetal growth restricted individuals, and that restoring insulin function in the brain completely reverts the behavioral phenotype in rodents. Now, I'm not going to go too deep into the role of insulin today, but as a brief side note, we have just published a review discussing how insulin enters the brain and modulates neurotransmission, acting on its receptors in the brain. And insulin, therefore, may be an important mediator of the effects of the earlier adversity. And in case you're interested, the reference is here. Now, as much as the animal models help us understand these mechanisms, one question that remains is how much these biological processes are relevant or are even happening in humans. How can we study variations in these biological processes, specifically in the brain, in clinical studies, that's in children cohorts, in the context of exposure to adversity, and associate these variations with the behavioral phenotype? My lab has been using functional genomics and polygenic scores to face these challenges. We know that genes are inherited and they don't change during the life course. However, small variations in the sequence called single nucleotide polymorphisms or SNPs are responsible for the differences in our phenotype. The variation that we see at the phenotype level, in fact, reflects variation in biological function at the molecular level. We all remember that the sequence of our genetic code in our DNA is the template used during gene expression to guide the cellular processes leading to protein synthesis and therefore controlling the cell machinery. From gene expression up to the actual function of a protein or enzyme, there are many levels of regulation or epigenetic process, but in fact, the template is the gene. So in a large human population, small variations in genotype will be linked to subtle variations in certain biological processes that will define the phenotype. However, genome-wide technology has been used so far with a high focus on disease state, rather than on the biological processes leading to that disease. By focusing on the comparison between two groups, phases and controls, genome is nothing like the subtle clinical variations and the phenotype manifestations that we see in establishment and diagnosis. And the clear statistical association between each genetic variant and the disease does not directly inform about the multi-layered and interactive biological mechanisms. So prompted by these issues, my lab has suggested a new way to calculate polygenic score, informed by biological function that we call expression-based biologically informed polygenic score. The scenario for our study in humans is this one. We are investigating the interaction between exposure to early adversity and individual variation in the dopamine neurotransmission in the mesocortical system. Depending on the exposure and on the way the mesocortical dopamine system works, individuals will have more or less risk to develop cardiometabolic and psychiatric comorbidities in adulthood. And hopefully we can attempt to identify the anatomical functional pathways associated with this risk using. So as I said, my lab has devised this new way of prioritizing SNPs, including polygenic score, and based on the principle of gene networks that considers that gene expression is co-linked by other genes, and consequently genes involved in the same network are expected to have similar expression profiles. In other words, genes expressed together work together in the same biological process. This is the bioinformatic pipeline leading to the calculation of these scores that we call EPS. I will spare you from the details, but the basic idea is to use gene expression data and sequencing to construct co-expression networks and identify genes that are working together with a gene of interest. We were interested in the genes that are co-expressed with the dopamine transporter, or that one gene, in the brain as a phospholimbic system. And the reason is because the dopamine transporter is a key molecule regulating dopamine as it controls the amount of dopamine available in the synaptic cleft and is the target for the mechanism of action of important therapeutic and abusive drugs. Note that the fact that this pipeline starts from the expression data, it is a brain region specific score. We then identify genes co-expressed with our gene of interest, map all the mix on these genes, and using genotype information from our sample, calculate a polygon score weighted by the association between our use and gene expression using the text. At the end, variations in the polygenic score will reflect individual variation in the expression of the gene. And in an attempt to capture more in terms of the complexity of environmental exposure, we use the adversity score as a method of early adversity that includes information on people growth, but also other variables like smoking or pregnancy, status, exposure to violence, etc. The outcome in the adults was the presence of comorbidity, defined by the diagnosis of at least one psychiatric and one cardiometabolic disease. And in the adolescent cohort alpha, the comorbidity risk profile was defined by the concomitant increase in depression and anxiety score, coma index, and waist circumference between the ages of 15 and 17 years. We observed significant interactions between the mesocortical index type 1 PRS and early life adversity and the risk for having psychiatric and cardiometabolic comorbidity in both adults and adolescents. The results also indicate a developmental trajectory for the risk to develop these comorbidities in adulthood, in which early indicators of this can be seen in adolescents as a function of the mesocortical index type 1 co-expression genetic and early life environmental conditions. And finally, to understand the anatomical functional relationships between genes and brain regions that could be involved in the phenotype presentation, we performed a parallel independent component analysis in the UK biobank. The results revealed that a subset of genes from our original gene network that were involved in synaptic plasticity, neurotransmission, and glutamate receptor activity are acting or playing together with brain matter content in regions like insula, the medial and inferior frontal gyrus, the striatum, and the parahippocampal gyrus, and that these relations could be involved in the modulation effect of the mesocortical type 1 gene network and the association between early adversity and adult psychiatric and cardiometabolic. We observed that the association between environmental and genetic factors can place risk for other comorbid chronic conditions from an early age, and that the dynamic transmission has a central role in moderating the effects of the early environment on the risk for the disease. These are examples of studies performed in my lab where we use a gene network by environment interaction approach to investigate child development. We also use a life force perspective for the identification of individual differences in responsiveness to adverse and supportive environments, and the integration of models, animal, clinical, observational, different data modalities like gene expression, and biomarkers, I would like to thank my student section that received it as soon as the PhD student leading the platform to work and the field WCNR. Thanks also to my collaborators, the funding sources, and you for your kind attention. Thank you.
Oh, okay.
There?
I think so.
Okay. So I had some slides prepared, but we're running a bit late, and I do want us to actually have you guys ask some questions. So I'm just going to talk, hopefully not taking too terribly long. This symposium reflects what I love about ISDP. We have animal researchers, human researchers, crossover researchers. This is one of the few societies where we really get a chance to work back and forth between those of us who study humans and those who study animals, and to really begin to force that conversation where you say, as I think Patricia said, by the way, I'd like to come back in my next life as Patricia. She stuns me. But as she said, you know, we can do all of this in animals, but will it apply to humans? And that back and forth and being, in a sense, disciplined by the human data, which is not terribly disciplined because we can't get in there and control everything. I wanted to point out a few things. One, it is very clear that there is crosstalk between BMI and elevated CORT. We actually saw that in the 3 studies that looked at it, and the 2 human studies were measuring cortisol in very different kinds of ways. So there's something maybe going on there. Megan, she's Megan, I'm Megan, I get confused. Focus on the hypothalamus and all the, you know, its plasticity and the impact that it may have, I think is absolutely brilliant. And Patricia's focus on the dopamine system, and read that paper where she says, beyond glucocorticoids, let's look at insulin. It is an incredibly interesting paper. You have insulin findings. She's got insulin findings. We need to look at insulin as a potential pathway. It's also very interesting that we see some very similar kinds of findings between those who are looking at maternal obesity, where the baby is likely to be big, and models of small for gestational age, where we're heading both ways up to problems with metabolic disease and disorders. So I just love seeing all of these coming together. And I feel sad for my students who study humans only, because it's really hard to get clean data. And the car, oh my God, the car, had I known you were looking, oh my God, the car is so hard to get a hold of. But the findings are there, and fascinating findings. And some of it will discipline the animal researchers, because you are seeing increase with low CORT, not high always. And so we need to figure that out. So how do I open this up for discussion? And can we bring our guys on? Our Mariann and Patricia on. We don't know what we're doing. Joan,
Is the table mic on?
the table mic can come on.
We'll do that.
There they are. So look a little frozen. Oh, Patricia, are you there, Patricia? Hey, okay.
Yes, hi.
Okay, good. I can moderate it.
That would be awesome. Thank you.
We have a student who's seen what's in the chat. Yes? Okay, let's start with a chat question. Is there nothing in the chat right now? Okay,
no,
be
it's all related to the audio issues. So no questions about science yet.
Our redhead over here. Who are you, by the way? Pull that mask down. Oh, it's you! Oh!
Hi, this is Susie Swithers. So I'm curious, you guys were talking a lot about obesity and overnutrition. And I'm always curious about whether this is related to obesity, BMI, or diet, because they could be potentially very different from one another. And of course, I can pick on Megan because you have an animal model where you have much more ability to explicitly control that. But that's kind of what I was starting to ask. And then Brie, while you're talking, I was thinking about, I didn't catch this, whether all of your moms were these their first pregnancies, and the extent to which their pre-pregnancy BMI could be affected by the fact that they may have had a previous pregnancy from which their BMI had not recovered. So those are two sort of.
Can you guys hear us?
Could we?
Do I have to push? Oh.
Whoa.
Okay. I think I turned this off, so we're not.
Good.
Okay, that sounds pretty good, right? Yeah?
Yeah,
Okay.
okay.
Do you want to go first?
That's a fantastic question. And we do have a lot more control in the animal world. Every time I venture into the translational world, even and dip my toes, I cry. But to answer your question, there have been several transgenic mouse models or gain and loss of function mouse models that have used standard chow diet, but just changed the body composition of the female pregnant dams, where you're just looking at excess adiposity in pregnancy. And then there's several nutritional diet models that are diet-induced obesity and manipulate the diet in a lot of different ways. So it's just a high-fat diet or just a sucrose diet, and the fat is different in the sucrose or fructose, or you can do all those things. They all lead to a phenotype of either babies being growth restricted or babies being larger in general. There's still variability, just like we see in human studies. And if you're born, it's similar to what Brie, I had a mentor in residency and fellowship that said the same thing, the Goldilocks phenomenon. If you're too small or too big as in your growth trajectory as a fetus while you're developing, that seems to be problematic. So I think from an adiposity body composition, the endocrine milieu is still off. There is something related to the adipokine profile of adipose and excess adipose, particularly leptin and adiponectin, that can cause differential effects in many organs, but in the brain. And then there is something with nutrient sensing that happens in the placenta and just the placenta structure and function itself that then changes what the fetus is exposed to and how they develop. So I think you are looking at, it's not going to be a one mechanism, right? It's going to be a network, which I get really excited when people are starting to talk about networks, because that's what it is. There might be a modifiable mechanism within that network that seems reasonable within pregnancy or outside of pregnancy in that plasticity place of the brain or some of this organ development that we can leverage based on some of this information. So that's the general.
Ask Patricia a question, and we'll come back to your end. So Patricia, you do reduced diets, so starving moms in essence, right? And yet you're seeing very similar pathways. I'm curious, would you expect that if Megan did responses to palatable food, she'd sort of see some of the same things that you're seeing?
Yeah, it's fascinating, as she said, that one of the reasons of being born small for gestational age is in fact maternal obesity during gestation, right? So, yeah, I think that the two ends of the spectrum, in fact, behave similarly in terms of the increased risk for disease in the long term. I think that there's an important difference on and one thing that she did not mention now in the response was what happens in the child after. So the child growth trajectory, not only growth in general, but specific tissues like the pancreas growth trajectory, and the adaptive responses to the environment that the child will face will also help to mold and to build this long-term effect that we see. So it's a huge, complex interaction between what happens before and what happens during birth, and how the child will evolve over time.
Yeah, I just want to agree with you. I mean, I think one of the challenges of a lot of prenatal stress studies is that our diet measures and nutrition measures are terrible if they're measured at all. And so, gestational weight gain and BMI are kind of one of those things that are easy to collect from chart review and seem to be in a lot of these prenatal. I mean, there aren't a lot of nutrition studies prenatally who have multiple measures of cortisol and stress right across gestation. So I would say that's a huge limitation. And I think I always cringe a little when using BMI. I didn't look at parity. I think most of them were, it was their first, but I can look into that and get back to you because that's a great question. But I think there's also in the human like model, I guess, this real challenge around one, that normal weight and kind of overweight for pre-pregnancy BMI is not associated with a lot of worse outcomes, right? Sometimes it's protective. And then on the other hand, I'm thinking of Mary Dahlman's work, where early life stress, like animals were actually using high fat, high sugar foods to downregulate their cortisol response behaviorally. And so I think diet can also be, really adaptive potentially in the short term if you are able to regulate your system in such a way, maybe when you're facing some pretty extreme stressors. So yes, it would be my call out to definitely get better, collaborate more with nutrition and diet folks to actually get better measures to look at how they interact across gestation. And thinking about Mariann's sample, where I'm very curious about that because it seems, and maybe Mariann can talk about this as well, but the high pre-pregnancy BMI and the high gestational weight gain seem to come to a cortisol level that looked normal, right, Mariann?
Sorry, I was just looking to unmute. Yeah, I mean, I think that's such a good point. And when I was watching my own talk, I was asking myself some of these same questions. I think what we don't know, especially, for example, about my cohort and your cohort, is like, what is the range of risk profiles? And so, when I'm saying we see these low levels of cortisol in reference to what? To the other women in this sample, I think that it's hard to draw these conclusions about linear models when we have been talking about curvilinear relationships, both in terms of cortisol and growth and BMI. So that's not a great answer to your question, but I think important to think about, like, what is the range of risk we have in our cohorts.
After 6, there's a cash bar, and you all have tickets for one. If you don't, you can get a ticket. So I'm just going to let one more question in. One more. Make it simple.
There's a question in the chat, actually.
Oh, there's a question in the chat.
Yes.
Okay.
So they said, what would you suggest if you were looking at diet in humans during pregnancy using diet calls? What could we conceptualize as nutritional, adverse, and human research?
Ooh. Oh.
You go for it. Yeah. I'll start with what I can control. What I can control. So we've looked at correlations between the fat intake and also the sucrose intake to try to parse that out a little bit. It seems at least in the animals, which obviously would be important to replicate in human cohorts, sucrose seems to be more detrimental to females in terms of hypothalamic kind of activity, and fat seems to be detrimental to both sexes in a lot of different ways. But
you've done diet recall.
Yeah, so I think just like the cortisol research, right, where there's so much individual variation. I think kind of more of the gold standard are 24-hour dietary recalls over multiple days captured over time. Although I will give a shout out, I think Jenna Doom is here, but she's actually working on where people take photos of their food and then use kind of coding and artificial intelligence to kind of quantify macro and micronutrients there. This is a great question from people from the chat. I think it kind of depends on your outcome of interest. I think fat and sucrose are, and sort of the insulin response system, right? Because there is individual variation there. I know. I mean, insulin is like fun and annoying at the same time, but you know, some people might have a really intense glucose response to a specific kind of diet or a specific kind of food that another individual might not, or might have a more normative response. And so biology, as Megan says, biology really wasn't here for us to study and understand. It was here to keep us alive. And so I think if we kind of take these things forward to try to understand what's going on, I think that's maybe the direction I would go. I would be something that Megan didn't talk about was that this was really a preclinical model of gestational diabetes, right? So she did mention it, but it's sort of this insulin response was sort of approaching this dysregulation, but not there quite yet. And so I think there's some interesting work to be done around just like glucose tolerance across gestation and stress. And you just layer diet. I think we just need huge teams of really excellent researchers who are interested in doing this in a lot of different perspectives. But yeah.
Well, I want to thank these two and the two on the screen and all of you, and let's give them a round of applause. And if you had a question, I'm sure you can find one of these guys to chat with. So let's go and try to get ready for the poster session.